Hello there. I'm Dr. Aubrey de Grey, the Chief Science Officer of Sense Research Foundation, and I'm delighted to be here hosting the session on aging at the World Stem Cell Summit. I would like to thank Bernie Siegel and his colleagues for inviting me to do this, and I would also very much like to thank Josh Hare and his colleagues at Longeveron for sponsoring the session. Here we're going to hear three very diverse and excellent talks. First of all, from Josh Hare. Secondly, from Dr. Jyothi Devakumar at Prime Movers Lab, talking about the investment perspective in the longevity space as of now. And finally, from Dr. Brian Kennedy of the Center for Healthy Aging of the National University of Singapore. I hope you will enjoy the session every bit as much as I will. Thank you very much.
Hello, everybody. My name is Dr. Joshua Hare. I'm very pleased to be participating in the session entitled The Longevity Session, Canceling the Pathologies of Aging. My talk title is Geroscience, Can We Address the Biology of Aging? These are my disclosures. And because Longeveron is a publicly traded company, here's the company's forward-looking statements. I'm showing this as I will be presenting some data from clinical trials conducted and sponsored by Longeveron. I think we all know that aging has biological hallmarks, opening the possibility for biological modification. This cartoon is a classic cartoon from Lopez Oten, and there are many variants of this cartoon. Some of the issues that have been outlined in these types of conceptualizations of the biology of aging have led to a new class of medications, particularly senolytics that address cellular senescence, but other ideas that we can pharmacologically or biologically modify the aging process. From the medical perspective, the goal of this is to achieve successful aging. I think we're all very familiar with the fact that life expectancy has increased, but health span has lagged. And there's over a decade of debilitation, particularly in the most advanced countries on the planet. In fact, the United States has the longest period of poor health span that is reaching about 12 years. What this means is that the period of life where we transition from having high and stable capacity to decreasing capacity and eventually substantial loss of capacity, in which we have many advanced chronic conditions and have a poor quality of life, is increasing and rather lengthy. This has been, I think, particularly clear in the COVID-19 epidemic, where it was very apparent that older individuals had the greatest vulnerability to the COVID virus and suffered the greatest mortality and greatest need for hospitalization. So eliminating or decreasing this period of decreased capacity or loss of capacity can have very substantial impact on the human being's quality of life and have major impact on the health of society in general. So the question becomes, can we apply biological interventions? And as I've already alluded to, there are a number of strategies currently under testing, including senolytics, metformin, rapamycin. But our group has been particularly interested for the last decade in applying infusions of mesenchymal stromal cells or medicinal signaling cells. The biological contributors to aging pathology can be distilled down to signs and symptoms and mechanism of action. Again, I think this is information we're all very familiar with, and it's intuitive that aging pathology is associated with diminished cognition, diminished cardiovascular function, immunosenescence, which refers to a diminution of the immune system ability to respond to pathogens or mount vaccination responses. And again, this was very clear in the COVID epidemic, that immunosenescence played a critical role in increasing the vulnerability of the elderly. This is driven by a pro-inflammatory state and is also has as a hallmark diminished strength and endurance and increased fatigue, which, as we'll come back to, are signs and symptoms of aging frailty. Now, the mechanism of action of this appears to be, amongst the other hallmarks, decreased stem cell activity, degeneration of tissues, and inflammation. It's for those reasons that our group hypothesized about a decade ago that mesenchymal stem cells, or mesenchymal stromal cells, which have a pleiotropic mechanism of action, could address the biology of aging. This schematic from a publication in 2015 highlights how mesenchymal stromal cells work. They have a very limited differentiation capacity, but a rather powerful paracrine signaling effect that's mediated through the release of proteins, but also cell-cell effects that involved the formation of nanotubes, mitochondrial transfer, actually heterocellular coupling, and importantly, the release of potent exosomes that are small extracellular vesicles containing a large wealth of microRNAs, mRNAs, and other peptides. Together, these effects can stimulate anti-inflammatory effects, anti-fibrotic effects, stimulate intrinsic regenerative repair, improve endothelial function, improve immune function, and improve mitochondrial function in the host. And as you could see, if you tie this back to the hallmarks of aging, you could see that this is a very, very plausible hypothesis that an agent that can achieve all of these things in the adult human could have an anti-aging or a pro-healthspan effect. The company Longeveron licensed technology from the University of Miami in 2014 to begin a program of work looking at this type of technology in aging-related conditions. Their product is called Lomecel-B. It is derived from allogeneic bone marrow, derived from young healthy donors who undergo stringent screening. These cells are amplified in culture in a cGMP laboratory and provide a high purity and viability MSC product that the company has termed Lomecel-B. This product has been tested in a large number of clinical trials now to test the general hypothesis of whether or not we can modify, or if you will, cancel aging pathology. Now, this is a chart from another publication in 2019. There's a lot of information here, so in the interest of time, I won't go through it in detail, but you see in the left column, there are pillars of aging that have already been mentioned many times: inflammation, stem cell and regeneration impairment, et cetera, et cetera. The MSCs have biological properties that can address all of these and therefore can address the hallmarks of aging. Now, I'll take you through a couple of examples. I think one of the most prominent examples is Alzheimer's disease, which is a severe form of cognitive diminution, and it can be viewed as cognitive frailty to the extreme. This is very prominently in the forefront in the past week with the announcement that Biogen finally, after about 20 years of research, got a monoclonal antibody conditionally approved by the FDA to address one of the pathological hallmarks of Alzheimer's. What you could see from the cartoon is that Alzheimer's disease involves severe atrophy of the brain. You see the healthy brain contrasted with severe Alzheimer's. There is a very substantial atrophy of the hippocampus in Alzheimer's disease, which is the site of new memory formation and emotional regulation. And individuals with Alzheimer's have very rapid decline in cognitive function and are debilitated very, very quickly, requiring institutionalization and having early mortality. Now, the FDA has not approved any cures but most recently, as I mentioned, approved a monoclonal antibody from Biogen, which reduces amyloid plaque, which is one of the key issues of the biology of Alzheimer's disease. However, as I've already mentioned, Alzheimer's disease is also associated with very significant neuroinflammation and tissue degeneration, particularly in the hippocampus. And so it's very unclear at this time whether just removal of amyloid deposition can cure the disease. And as a result, our group hypothesized that infusions of mesenchymal stromal cells would potentially improve cognitive function in patients with severe Alzheimer's. This cartoon slide addresses many of the underlying biology of Alzheimer's disease. There's the amyloid issue, as I've already mentioned. There's also a mitochondrial hypothesis, dysfunction in the vasculature, a metabolic hypothesis, and all of this is driven by neuroinflammation. There's been a very substantial amount of preclinical studies looking at MSC-like cells that show very substantial improvements in cognitive function in mice models, driven by improvements in inflammation in the brain, vasculature, and a promotion of neurogenesis and differentiation. This is the very recently released data from Longeveron about the trial that they conducted. This was a phase 1 study. However, it was done in a randomized fashion in order to achieve exploratory information for future trial design. And the very exciting results of this trial were that in one of the arms of the trial, and subjects had received 20 million lomacel B cells infused intravenously compared to placebo, showed significant evidence of slowing cognitive decline. You see the reduction in the mini-mental score in the placebo group. It's very substantial, greater than 5 units in 52 weeks. And you see how this is substantially decreased with lomacel B. A similar trend can be seen with the ADAS-Cog, in which an increasing score represents worsening. You see the placebo group worsens and has increased by about 5 points at 6 months. And here again, this is slowed by lomacel B. Now again, this is small, a very small study with a small amount of data, but it's absolutely going in the right direction and is going to form the basis for an upcoming much larger clinical trial. It certainly speaks to the idea that cells like lomacel B can modify aging-related pathology. Now, if lomacel B can do that in the brain, perhaps it could do that in the whole body and address aging frailty per se. So again, these pleiotropic therapeutic effects to target and reduce inflammation, improve immune function, stimulate endogenous repair, not only in the brain, but perhaps in the skeletal muscle and in the cardiac muscle, can achieve an improvement in aging frailty. My group at the University of Miami conducted a phase 1 and 2 study, the CRADA studies, which were published in 2017, that again revealed exciting and provocative information that lomacel B, or a product that is similar to it, made at the University of Miami, was safe to infuse in these patients, and indeed improved strength, balance, and endurance, enhanced cognitive function now in individuals who don't have Alzheimer's disease but just have aging frailty, improved lung function, had a powerful systemic anti-inflammatory effect, improved cardiac and endothelial function, and potentially boosted the body's ability to fight and prevent infections. This data in the CRADA study formed the basis for a much larger program of work conducted by Longeveron. Now, one of the studies that is nearing completion is a study in which the immunosenescence hypothesis was tested in older individuals. First of all, I've already alluded to the fact that older people are much more susceptible to viral infection and also have a diminished response to vaccination. This is because there is immunosenescence in the B cell repertoire and a much more difficult opportunity for the B cell repertoire to respond to a vaccination. So this is part and parcel of aging pathology. And a study was conducted called the Hare study, in which individuals were first infused with Lomecel B and then received vaccination. And you could see under two treatment paradigms here, you could see the immunosenescence in one of these groups, paradigm 2, in the red curve, and how this is corrected in individuals who got an infusion of Lomecel B at the right time interval prior to the vaccination. This preliminary data is also very exciting. It suggests that Lomecel B, through the mechanisms of action I've already discussed, can enhance the B cell repertoire and perhaps reverse immunosenescence. One of the critical drivers of all of the pathology I've talked about is an elevated level of inflammatory cytokines. And here is some data from the CRADA studies that looked at TNF alpha levels, which were elevated in this patient group of older individuals above 5, not markedly elevated, but certainly way above normal, which is about 1. And you could see that the infusions of Lomecel B, in this case 2 infusions given sequentially each 6 months, showed a progressive and sustained decline in individuals over a period of more than a year. This type of data is, I think, very important for the mechanistic understanding of how infusions of cells like Lomecel B could reverse aging pathology. So just to sum up, I've already talked about the signs and symptoms of aging, the diminished cognition, the impaired cardiovascular function, the immunosenescence, which I gave you a nice example of from the Hare study, the pro-inflammatory state, the diminished strength, endurance, and increased fatigue, the hallmarks of frailty, due in part to decreased stem cell activity in the body and an impaired offsetting of the natural amounts of tissue degeneration that we all experience with age. The potential benefits of Lomecel B are to improve cognitive function. I showed you a very dramatic example from Alzheimer's disease, where a marked decrease in cognitive function over 5 points in the mini-mental score over 1 year was greatly diminished by a single infusion of Lomecel B. I've alluded to the improved lung function we showed in the CRADA study, improved cardiovascular function, the improved immune system in the vaccination data, which, by the way, is going to be tested in a new study that's completed enrollment and will be announced soon. The sustained decrease in inflammation and improvements in musculoskeletal function that I didn't have time to talk about at length in this talk. I will tell you that Longeveron, which was formed in 2014 and recently has gone public on the NASDAQ, has a very, very ambitious program, as highlighted here, international in scope, looking at neurologic diseases, aging frailty, cardiovascular disease, and inflammatory diseases, and is conducting a large number of trials and funding partnership with the Alzheimer's Association, the National Institutes of Aging, and TEDCO, a fund from the state of Maryland. Longeveron also has a treatment registry in Nassau, the Bahamas, which has also provided the company an opportunity to treat some very, very difficult cases under an expanded access approval received from the government of the Bahamas. So just to sum up, the question is, can we cancel the pathology of aging with biological interventions? I would turn that around to the geroscience hypothesis and say, not only can we, but we must attempt to favorably modify the biology of aging, because it will have profound positive effects on the quality of life of individuals that are now experiencing a very poor quality of life in the last decade or plus number of years of their lives, and also offset the dramatic healthcare burden that's now imposed on societies that can be attributed to the increasing number of aging individuals. Thank you very much for the opportunity to participate in this session. I have many individuals to acknowledge, particularly the staff at Longeveron, a long list of collaborators at the University of Miami around the country, and very importantly, acknowledgement of the National Institutes of Aging that's provide Longeveron an R44 and an R42 grant. Special thanks to the Alzheimer's Association that funded Longeveron's Alzheimer's project with the Part the Cloud grant, and a number of awards from the Technology Development Corporation of Maryland. Thank you very much, and I look forward to the question and answer session.
Hello everyone, my name is Jyothi Devakumar. I am the principal biologist and partner at Primovers Lab, a venture fund that invests in breakthrough science that can impact billions of lives. The focus of this presentation is to take a look at the field of longevity from an investor's perspective. What makes this industry relevant for investors? Is this still a faraway dream? Is it even worthwhile the investor's time in taking a look? The attraction of longevity as an investable industry makes sense from several different perspectives: the demographic perspective, the biomedical context, and the commercial context. The statistics of the gray economy is very gloomy, but the silver lining is the opportunity that it provides from a technological perspective and the possibility of saving billions of lives, which can translate into trillions of dollars, makes this one of the most coveted investment areas. The combination of falling fertility rates and increased life expectancy is changing the population age profiles. Issues related to increasing number of people suffering from chronic age-related diseases is capturing the attention of policymakers and changing the mindset of public to aging. Some of the numbers are depicted in this slide. I'm not going to go through all of these, but I would like to highlight just one aspect of the slide, which is in 2017, the proportion of people aged 65 and over was 1 in 8. And this is estimated to increase to 1 in 5 by 2050. Unfortunately, some of the countries are already in this range of 1:5. For example, Japan leading the tally, Italy, Greece, and Portugal. Now let's look at the longevity as a valid sector from a biomedical perspective. Before getting to that, we need to understand some of the hypotheses and developments that have propelled this industry. The first one is the understanding that most of the chronic diseases have a common factor, which is aging. Lifelong accumulation of damage drives aging. This, in fact, has become the de facto definition of aging. No underlying genetic program that defines the maximum lifespan. Several studies in animals have extended lifespan. For example, in C. elegans, some of the interventions have doubled the lifespan. Identification and classification of hallmarks of aging. I'm going to briefly touch upon that in my subsequent slide. Concept that lifespan versus health span, and the understanding that health span extension is critical. Technological advancements today are making this goal realizable. What is the impact of increased lifespan? One, there's decrease in healthcare costs, insurance expenditure, and also increased work span. One study by PwC illustrates this in a very beautiful way. They looked at the UK, and based on their estimates, one increase of 1 year in work span can increase the GDP by 1%. So that should tell you the impact of increasing health span. Now let's quickly walk through the hallmarks of aging. As I mentioned, hallmarks of aging is a dominant concept in the field, every conference on aging has people presenting this over and over again. And to me, hallmarks are molecular roots that are shared by most, if not all, diseases of aging. Scientists believe in a varying number of hallmarks, and the one depicted here is from one of the most cited papers. What are these hallmarks? One is DNA damage. This is caused by mutations, epimutations, retrotransposons, all arising due to imperfections in repair mechanisms. The second hallmark is that of telomere attrition. These are the ends of chromosomes that continue to decrease with each cell division, leading to arrest of cell division, which in turn pushes the cell into one of the following states, maybe apoptosis, cancer, or senescence. Changes in gene expression patterns that are out of sync due to aberrant epigenetic regulation. Failure of adequate turnover, leading to accumulation of unwanted proteins in the cells, forming aggregates, complexes, which could be toxic. Malfunctioning of the body's energy production and control machinery, which is namely mitochondria. Loss of ability of cells to sense and adapt to nutrient availability. Cells losing control to become zombie cells, popularly called senescent cells. The regenerative pockets in the body contain cells that can repair, replenish, and replace, which either lose their ability to function effectively with age or diminish in number, leading to compromised ability of the body to heal. Lastly, there is altered communication between cells and also altered extracellular matrix. Let's then go on to look at the commercial context. Why is there a continued increase in commercial interest? For decades, the entire drug discovery industry focused on developing drugs for diseases, as aging was not considered a disease. And even if they did discover drugs to combat aging, the regulators simply would not approve them because there was no pathway whatsoever. No industry, including the drug industry, can sustain making products that they cannot legally sell in the market, or there is no market available. Now, with the changing demographics and mindset, the aging is not a disease problem is going away. And all this is summarized by a beautiful quote by Bank of America, which states that in 2019, the market estimate for longevity was 110 billion, and this is expected to increase to 610 billion, representing one of the biggest opportunities in the coming decade. How is the industry getting away from the mindset of drugs for diseases to drugs for aging? There are various methods the longevity sector is adopting. One, by initially positioning the broad spectrum anti-aging drugs to a specific age-related disease to obtain the regulatory approval. The well-known TAME trial endpoints are all related to aging, but without use of the word aging, thus avoiding the need to have an agreed definition of the word. The endpoints are all individual conditions that are associated with aging brought together in a correlated manner in the context of comorbidities. In addition, WHO has classified chronic diseases within the ICD-10 by adding an extension code, age-related. And this gives the required elbow space for the practitioners to prescribe drugs to the aged population. And also, because this is such an emerging sector and it's in an early stage, there is minimal competition. There's a huge collaborative environment leading to several co-investment opportunities and enthusiasm. This is something we come across every day as a part of our investment world. How does this translate on the ground? Have we made a case from the demographic, biomedical, and commercial perspective? We have also looked at the ways in which the regulatory hurdles are being addressed. Are we making progress in the industry? Yes, there are plenty of examples to show the progress. For example, look at the field of senescence. There are different types of companies addressing this problem from senolytics perspective, that means inventing drugs that kill these senescence, cells, there are xenoblockers, then there are xenomorphics, and then in the telomerase space, there is episomal modalities to increase or activate telomerase. There are drug-like molecules. Some of the interventions are nanoparticle based, and there are mitochondria based interventions, which include uncouplers that decouple the energy production from the membrane potential, which can potentially be the answer to the increasing pandemic or epidemic of obesity. Then there are functional stimulators. There's also gene therapy approaches. Then autophagy, including the chaperone-mediated autophagy, which is becoming the center of attention. All these interventions, irrespective of what they are, have common disease indications they're looking at, which include idiopathic pulmonary fibrosis, PCOS, type 2 diabetes, obesity, cancer, neurodegenerative diseases, to name a few. Now, no presentation on aging in the present time is complete without the mention of COVID-19. This pandemic has had a devastating effect both from a humanitarian as well as from an economic perspective. Though there are a large number of diseases that target the elderly, COVID-19 has captured the imagination of everyone, including policymakers, politicians, and the public. COVID has affected the elderly much more than any other age groups. It's also affected the chronologically not so old groups that have comorbidities usually associated with aging. It would really be interesting to understand the biological age of these affected groups, especially chronologically not so old groups that have the comorbidities. The pandemic has brought to the fore the importance of aging research, especially the importance of immune rejuvenation, the importance of naive T cells, and the organ that makes these naive T cells, namely the thymus, that involutes and becomes non-functional with advancing age. The need for improved strategies for developing vaccines and adjuvants that work well in the elderly has gathered interest, and every one of the vaccine companies is now using this approach, and the vaccines developed are differentially tested to prove their efficacy in the elderly. COVID-19 has highlighted the importance of longevity industry and the critical need for support both from federal grants as well as from investors. Let's talk quickly about the current longevity ecosystem. There are different types of investors. One main category here is the holding companies. These are the companies that take a majority stake and actually take a project all the way from formation of the company to its clinical endpoint or commercialization. These include Regenerative out of Switzerland, Juvenescence, LifeBio, Cambrian Biosciences. There are longevity-specific VCs such as the LongeVC, Longevity Vision Fund, and Longevity Fund. Increasingly, as the industry is gathering importance and visibility, VCs who were previously investing in other sectors are expanding their portfolio into longevity, and we come on Prime Movers Lab, the company that I'm associated with, is one such entity. And there are other ones, very important ones, such as Bold Capital, Khosla Ventures, etc. There are family offices that are longevity specific, like Kitsu out of Germany and Formic Ventures. There are family offices that have an extended portfolio but are increasingly showing interest in longevity, such as Lauder Partners. Now, with all these developments, how have we done on the ground? So I have cited 3 examples, and I'm going to lay my own set of optics on how to interpret these developments. Let's begin with Unity. Unity may seem like a cautionary example, but actually it offers great encouragement to the industry. They suffered a failure of a key clinical trial, phase 3 clinical trial, which impacted their share price greatly in the near term. But they've survived and recovered, interestingly, both due to the underlying strength of the technology and the sophistication of the investors who were able to see the trial failure as an isolated blip that it actually was. Secondly, in some of the other two examples that I have quoted, the gains might seem too small. For example, Alkahest, it's only $250 million. Now, instead of interpreting it overly, I think we have to aptly remember here how young this sector is. We as investors are always looking for the next set of unicorns. As the wisdom goes, looking for unicorns in an emerging, fast-growing field is much more rewarding, and longevity is precisely in that stage and has several such unicorns to offer. And Sensei has gone through an IPO in early 2021 despite all the issues the world had because of COVID. It's an immunotherapeutic company which is with a focus on cancer, and very soon some of the candidate molecules from Sensei would be moving into phase 2 clinical trials. We as a fund look at companies in the sector on an everyday basis. We also understand how the companies we see in this space compare to other sectors that are much more mature. In the longevity sector, we do see bottlenecks, as in the case of any other emerging industry. Here I'm broadly classifying them into 3 categories. First, let's look at the seed stage companies. Most of these are formed by professors who would have spent all their lives in academia. They go through several pain points, such as no defensible IP, maybe because the company was spun out a tad too early, dissociation from the university making access to well-equipped labs difficult, maybe, and need for commercial support to run the startup. Just sheer lack of commercial knowledge here. Once the companies are able to establish and transition into Series B and beyond, issues related to standardization, especially from the biomarker and functional readout perspective needed for IND studies, come into picture. Efforts are constantly being made by industry associations and consortiums to resolve these issues. We've already discussed the regulatory bottlenecks, but the picture is getting better each passing day. Now let me quickly walk you through who we are. What do we do at Prime Movers Lab? We scale breakthrough scientific startups funded by prime movers, the inventors who've transformed billions of lives. And we treat founders like customers rather than as mere investments. We deliver a mix of engineering advice, coaching to scale, critical hiring, and support for commercialization. We have a bunch of PhD level scientists and best-in-class technical advisors, and we have in-house we have 3 funds. Fund or Lab 1 fund, which has yielded 8.6 times revenues in the last 3 years, is a $100 million fund which has already been deployed. The second fund is the fund Lab 2, which is a $245 million fund which we are deploying out of. And then we've closed a growth fund on a $375 million target and expected to hit its $500 million cap shortly. We have our founder is Dakin Sloss, a serial entrepreneur and an established venture capital expert. And our thematic areas of focus include transportation, infrastructure, energy, manufacturing, and agriculture. And the most important one for me is human augmentation. Under human augmentation, we have 3 broad areas again. One is longevity, cognition, and enhancement. Human augmentation and longevity is our expanding portfolio. So far, we've invested in 4 different companies that roughly fall in this category. One is Guardian. It's a gene therapy-based drug discovery platform which allows testing of multiple variants and their combinations in a single setting. And then we have Elevian, which is focused on GDF11 and its regenerative capacity that mimics that of the young blood, and it has multiple applications. Then iViva is the next generation engineered functional tissues, engineered functionalized tissues, and we have Vaccinity, a vaccine development company with a platform technology and can develop vaccines that work in the elderly. That's my investor perspective on the longevity industry. Thank you very much.
Thank you for inviting me to talk today about human aging. I'm going to tell you first a little bit of the studies that have gone on in animal models that have led us to believe that it's possible to extend human lifespan, and then, more importantly, how we're going about validation approaches to make sure that human health span can really be extended. Now, if you look on the left here, you'll see a bunch of different diseases. And clinicians have typically considered these to be separate. You don't typically go to the doctor's office while you're healthy. You get sick of one of these diseases, and then you go, and then there's a lot of effort made to try to treat these diseases. And even research institutes have failed to recognize the things that are in common between all of these chronic diseases. If you look at the panel more on the right, I think it makes more sense. There's a risk factor that drives almost all of these diseases, and by far the biggest risk factor is aging itself. And this leads to a relatively simple concept that if you could slow down the aging process, it would be possible to delay or prevent the onset of all of these chronic conditions at the same time. Now that seems like a big goal, but actually I would argue it's not big enough, and that's been reinforced to us over the last couple of years with this pandemic. Because it's not really just chronic diseases that are being driven by aging. It's functional decline, but also the severity of infectious disease. We know by far aging is the biggest risk factor for mortality when you get diagnosed with COVID-19. And that's also true of influenza and a variety of other acute infections. So that means that if we could slow aging, we could meet three major medical needs and try to live a longer, healthier life. So this is essentially what we're trying to do here. If you imagine this top timeline here, a person's born. In the green, they're still healthy and functional, but that slowly declines. They cross some threshold where they can't maintain homeostasis anymore, and they get disease. And this red period is when we try to treat people when they already have chronic illness. And we're not as good at that. What we do is we rarely cure these diseases, but we do keep people alive longer. And so in a sense, we're not doing health care, we're doing sick care. What we'd like to do is develop interventions late in this health span period, or in other words, middle age, where we can introduce into populations and extend this period of health, extend health span, and also compress the red period or compress morbidity. So the net effect of this might be that there's a little bit longer lifespan, but there's a lot longer health span. And the question is, how do we keep making this triangle bigger and bigger? Now, there's been a lot of work on the basic pathways that govern the aging process. Two major papers came out in about 6 or 7 years ago, one on hallmarks of aging and one on pillars of aging. They overlap a lot in what they talk about, but the main thing to take away from this is that all of these pathways are connected to each other. And so you can look at aging as altered metabolism, altered proteostasis, reduced stress adaptation and resilience, increased inflammation. But the truth is that there's a lot of connectivity. And I think a better way of looking at aging is that there's a homeostatic network that's in place to keep you healthy. And even though damage and things happen as you get older, that network compensates and you maintain function and you keep yourself free from major diseases. But eventually you cross this boundary where that network can't work anymore. There's too many problems. And then when it breaks down, that's when you enter this period where you get chronic diseases. And depending on your genetic risk factors, your lifestyle, you may get one type of disease or another, but it's that homeostasis that if we can preserve will keep you from getting all of the diseases. Now recently there's been a lot of work on cell senescence, and I don't have too much time to talk about today, But cell senescence fits into a number of these paradigms, and it may be one of the things that helps link them together. So none of this would matter. We can talk about slowing aging. People have been talking about that for thousands of years. But it doesn't matter unless we can do something about it. But actually, it's a misnomer to think that it's impossible to modulate your aging process. We already know pretty much that lifestyle interventions slow aging. Things like exercise, healthy diet, are pretty much shown to work, at least for extending the median life expectancy of humans. And then things like stress management, improving sleep quality are also likely to work, although there's less data on that to date. So that would be this dark red circle, lifestyle interventions. But there's an intermediate circle where a lot of research is being done right now, and that's on small molecules, supplements, drugs that are likely to slow down the pathways of aging. And I'm going to tell you a lot about that today. And then there's a broader circle which involves things like gene therapy, stem cell replacement, and a variety of other ideas that I think may ultimately have even larger effects than the first two circles. But right now we know much less about them, and they're probably not really ready for prime time, at least at the moment, in humans. So these are a variety of small molecules that we work on in my lab. If I listed all of them that are under study for aging research, we would be talking in the neighborhood of 50 or 100 even small molecules that may affect human aging. I'm happy to talk more about this. You can reach out to me if you're interested in specific molecules, but I don't have time to go through all of them today. Rather, what I'm going to do is to focus on one, alpha-ketoglutarate. And the reason we got interested in alpha-ketoglutarate was a company, Ponce de Leon Health, sponsored research in my lab and in Gordon Lithgow's lab when we were at the Buck Institute to try to identify combinations of small molecules that impact the aging process. And so this was first done in worms in Gordon's lab. And then once we found combinations that looked good, we studied them in mice. And the way we did that is by initiating interventions at 18 months of age and following the mice all the way to natural death, looking for interventions that extended lifespan. But remember, it's Healthspan we're interested in. And so we also used the 31-point frailty index introduced by Susan Howlett to try to get a better idea of the health of these animals as they're getting older. We didn't just look at that either. We looked at inflammatory markers, adult stem cells, and a variety of other properties of aging. Now, I'm not going to tell you today about the combinations that affect aging as much as I am one molecule, and that's calcium alpha-ketoglutarate, because it really had the most robust effect in these animal models. And I want to show you a little bit of animal data just to get you on board with how we're thinking and what might happen in humans. So this is the intervention at 18 months of age. It's a relatively small effect on survival. 10% extension in females, statistically significant, 5% extension in males. Didn't quite reach statistical significance. We now have 4 cohorts of mice with alpha-ketoglutarate, and we pretty much see these results all the time. And if you combine the cohorts, the males are significant, but the effective size is a little bit smaller than the females. Now, you wouldn't necessarily get super excited about this data. If you look at an intervention like rapamycin, the effect on lifespan is much larger. But what excites us about alpha-ketoglutarate is this slide, and this is a bit of a complicated slide. What we're doing here for each of these circles is measuring the frailty of an animal at a different part of their lifespan. So 18 months, 21 months, 23 months, etc. The total frailty index is a composite of all those 31 parameters, and the higher you score, or an animal scores, the more frail it is. This frailty index is quite nice because it has a relationship to human frailty measures as well. So what we find in mice may well be conserved in humans. But if you look at the frailty over the months, this is a bit confusing. You can see that the frailty goes up in the control animals faster than the AKG animals, and that's significant. But it's also hard to interpret, because if you take this animal here at 23 months that had a very high frailty, it was dead at 25 months. So it's not on this part of the curve. So what we did is to go back and replot the data, looking at the percentage of the animal's lifespan where we did the frailty measurement. So 50 to 60, 60 to 70, 70 to 80, 80 to 90, 90 to 100. And then when you do that, you see a relatively linear increase in frailty in the control animals, and that's substantially dampened in the alpha-ketoglutarate treated animals. So while we're only seeing a 10% extension of lifespan, we're seeing a 50% reduction in frailty. And we'd like to argue what this means is that we're compressing morbidity in these animals. If you imagine that timeline, we're extending the green and compressing the red. I think the appearance of animals is always worth looking at, and you can see that at 27 months, the alpha-ketoglutarate treated animals look much better than the control animals, which are on the right. Their coat condition looks better, they have less kyphosis, alopecia, really a number of parameters look better. These mice are just doing better and healthier. Now, what is alpha-ketoglutarate? Well, if you took biochemistry, you'll realize that this is a TCA cycle component, central metabolite. Right here it is right here. And it's an interesting molecule because it links carbohydrate metabolism to amino acid metabolism, because alpha-ketoglutarate is an end product of amino acid catabolic activity. And so this is really a molecule that's important for metabolic flexibility. It allows cells to have crosstalk between different nutrient sources. And it's an important molecule in that sense. But it also participates in hundreds of different chemical reactions in the cell. And that makes it a little bit challenging when you're trying to understand why it affects aging. Which enzymes does it act as a catalyst for, a substrate for, that it facilitates, that is important for longevity? And we've been able to show that a lot of pillars of aging are influenced: improved adult stem cell function, improved mitochondrial function, improved metabolic flexibility. We have very interesting data now we're starting to interpret on an altered microbiome. It essentially makes a younger looking microbiome. Fairly dramatic reduction in inflammation and effects on aging biomarkers as well. This is the inflammatory data, and I don't want to take too much time. But what you'll see is that for all these inflammatory cytokines, we've normalized the levels at 18 months of age in the mice in this orange color. And you can see that things get redder at 28 months in the control animals. And that means most of these inflammatory cytokines are increasing in levels. And that's substantially suppressed by alpha-ketoglutarate treatment. This is a fairly robust anti-inflammatory effect on these mice. And that could be one reason they're having an impact on their lifespan. But again, before I go on, there are many other factors that could be influencing the lifespan of these mice as well. And we're trying to look into that and to tease all of that information apart. I want to move on to our studies in Singapore, where we're looking at healthy longevity in the clinic. This is our new clinic where we're trying to measure aging and look at interventions to affect human aging. And I'll tell you a little bit about what we're doing. So you probably know this already, but it's worth re-emphasizing. We all count our age in years or chronologically, and our passport measures that very, very well. But you and I both know that people age at different rates. So if everyone aged at the same rate, it would look like this dotted line. And eventually you cross some disease threshold and start to get chronic diseases of aging. But we all know people that seem to age more rapidly and they get sick earlier. And we are lucky people also that age more slowly and they make it to a much later age before they get sick. So this is your biologic age. And now there are pretty good ways to measure the biologic age of an individual. And that's great because it means that we can test interventions and see how they affect these biologic aging parameters to validate whether they work or not. Now there are a whole range of, sorry, this slide didn't come out perfectly, but there are a whole range of biomarkers that we're looking at now to try to measure biologic age. The gold standard really is DNA methylation. So the methylation of about 300 sites in your genome can predict your biologic age. We're looking at autoantibodies, telomeres, inflammatory cytokines, cell senescence, as well as a number of classic physiologic parameters of aging, like physical activity, frailty, body composition, and cardiovascular function. I will also mention that there's a biomarker developed by Jackie Han in Beijing looking at biologic aging based on a three-dimensional reconstruction of your face, and we're comparing that to some of these other biomarkers as well. What we'd like to do in this is, by looking at all of these biomarkers, find the unique features that each of them tells you, but also the redundancies, because what we'd like to do is to develop a biomarker of aging that we can do for a few $100s, something we can scale into an entire population like Singapore and measure who's aging well and who's aging not so well when people are in their 40s or even 30s, so that we can develop interventions earlier on to keep them healthy. This is just an idea. So this is the DNA methylation clock, which is probably the most well known. Steve Horvath is certainly one of the people that's really driven this forward. And it's essentially looking at DNA methylation throughout the genome and using artificial intelligence to try to find which sites in the genome predict biologic age. And it works something like this. You get a methylation average over time, and you look at people's biologic age compared to their chronologic age. So at 60, this person chronologically may score as 65 or 70 or 80 even biologically, so they're not aging well, whereas someone else might be aging quite well still at 60 years of age. And this measurement really provides a way to test that. And so what we're doing is taking relatively short-term approaches, either healthy individuals or people that are near healthy, so they may have disease risk factors, but they don't have multiple overt diseases, measuring their biologic aging, and then looking at a range of different kinds of interventions to try to figure out which ones affect these biomarkers. And we're taking an agnostic approach. We're going to look at multiple different kinds of interventions and maybe even combinations of interventions to see which ones work best. And that could include diet changes or exercise, but we're also looking at repurposed drugs, supplements and micronutrients, and ultimately we want to look at things like stem cell injections to see really what works in human populations. So just to give you an idea, two of the first clinical studies that we're involved in are with alpha-ketoglutarate, since I talked about it already today. This is a study sponsored by the company on top, and it's being conducted independently of us by Indiana University, looking at the product. And this Rejuvenate product has not only alpha-ketoglutarate, but vitamins in it that are specific for men and women. The idea, though, is to take 45 to 65-year-olds, look at a 9-month intervention, and measure aging biomarkers and inflammatory markers. This study should be completed in the not-too-distant future. It got affected by the pandemic, but we're making a lot of progress on getting it completed now. So we're very excited to see what this data looks like. We're doing a study that's similar at Singapore, at National University Singapore, but here we're looking at just the calcium alpha-ketoglutarate because we want to get more mechanistic as to what the specific molecule is doing without being confounded by adding the vitamins. Again, though, relatively small study, 6-month intervention, 45 to 65-year-olds, and not just aging biomarkers this time, but immune and metabolic markers and a variety of other things. Really all the things I showed you on that prior slide will be what we're looking at. So this is the kind of thing we want to replicate with multiple interventions and work back and forth, still using preclinical models like mice to try to optimize what works best and how to measure it. And this is sort of the roadmap we have. We're at this stage now doing sort of in-depth profiling, looking at interventions, trying to convince ourselves which ones are likely to work. These will generate, I think, very interesting findings and hopefully a lot of papers, but we're ultimately going to have to go to randomized clinical studies or RCTs that are large enough to get regulatory bodies to pay attention to the observations that we're making. And then ultimately in Singapore, we want to go to community translation. How do we take interventions that extend health span and move them into large populations so that we're moving the health span needle rapidly on an entire group of people? And I think Singapore is a great test case for how to do this because it really has, it's a small island, there's great medical care, the government's already thinking about the issues surrounding aging. And one of those, for instance, is that there will only be 2 workers for every retired person in 10 years from now. So there's a huge economic challenge accompanied by the aging population in Singapore. And if we can keep people healthy longer, it will not only save lots of money, but it will dramatically improve life quality. And with that, I want to stop and thank you again for allowing me to give the lecture. And hopefully we can start doing these things in person again soon. Thanks a lot.